This is our last lecture to wrap up Chapter 2 on Sections 3 and Section 4 of Chapter 2. Specifically, what we'll be covering in this lecture is we will be reviewing acids and bases, so we're going to talk a little bit about what they are and also how we can measure how acidic or how basic a solution is using the pH scale. And then most of our lectures are really going to focus on the 4 macromolecules that are required for life. If you remember from our first lecture video from Chapter 1, all life has to have these 4 molecules: carbohydrates, lipids, proteins, and nucleic acids. And so today, and again, because molecules are truly kind of the smallest or simplest level of biology, we're going to be looking at these 4 molecules. We're going to be looking at kind of how they are made, but also some of their defining structures and functions. So a little bit of just kind of basic chemistry and then really focusing on those molecules that are key for life. Just as a reminder, Section 2.3 also covers water, and you should have watched that YouTube video from the Amoeba Sisters for that information. So I'm not skipping that, it's just I gave it to you guys in a different way. So let's talk about acids and bases. So we've probably heard the terms in kind of our day-to-day life that, oh, that's acidic or that's basic, but what actually are these in terms of chemistry? Well, both of these types of compounds are compounds that when they are added to water, they dissociate. What that means is that the compound, that's the bonds kind of between the atoms, fall apart. Acids typically dissociate, and when that compound falls apart or dissociates, it creates hydrogen ions, which is often denoted as H+ here. Bases are the opposite. Bases, when they are added to water, they dissociate and create what is called hydroxide ions, or OH-. So hydrogen ions are positively charged, hydroxide ions are negatively charged. What's important to know about both of these ions is that they are very, very reactive, and they can kind of wreak a lot of havoc or kind of destruction inside the cell. And so being able to determine how acidic or how basic a compound is can be really important to understanding kind of human health. So the way that we measure acidity or basicity is by using the pH scale. So pH specifically measures hydrogen ion concentration in a solution. So it tells us if we have high levels of hydrogen ions or low levels of hydrogen ions. It also allows us to compare how many hydrogen ions there are versus hydroxide ions in a solution. The pH scale is shown here on the left, and it actually kind of starts in the middle. So it actually starts here at a pH of 7, and it goes all the way down to 0 here at the top, or up to 14 at the bottom. So the pH can range anywhere from 0 to 14. A pH of 7 is considered neutral. What that means is that when something is neutral, the ratio of hydrogen to hydroxide ions is equal. Or we can say that the hydrogen ion or hydroxide ion concentrations do not change. Typically, like examples of neutral solutions are like water. Our blood should also be fairly close to neutral. It's about a pH of 7.4, so it's fairly close to neutral. Notice that as we go towards the top of the pH scale, our numbers decrease. So we're in numbers that are less than 7. These numbers are considered acidic. So anything less than 7 is considered an acid. That means that when these things are added to water, they will dissociate into and create hydrogen ions. We can also say that acidic solutions have higher protons or hydrogen ions compared to hydroxide ions. And some common things are like urine, beer, battery acid, lemons, tomatoes. Those are all going to fall in the kind of acidic range. As we increase in number, as we go from anything that's greater than 7, those compounds are considered basic. This means that when these compounds dissociate in water, they will create hydroxide ions, or another way that we can say this is that they have a lower hydrogen ion concentration compared to hydroxide ion concentration. And a lot of like cleaners that we use day to day to clean our house tend to be basic. So things like ammonia, oven cleaner, baking soda, those kind of are some common basic materials that we might run into in our day-to-day life. Something that I want to point out about this pH scale is that these changes in numbers as we go from 0 to 1 to 2, this is a logarithmic scale. And so what that means is that when we change pH as we go from, say, 7 to 6, those number differences represent a 10 times increase or decrease in acidity. So for instance, let's just say we have, if we're going to start out at a pH of 7, let's say we add something to our solution that gets us to a pH of 5. So we have 7, 6 sits in the middle, and then 5. That means that we have to move 2 units to get to 5, but each time we move represents a 10 times difference. So as we go from 7 to 6, 6 is 10 times more acidic than 7. As we go from 6 to 5, the pH of 5 is 10 times more acidic than 6. So if we wanted to compare of 5, if we want to compare these two units, we would need to multiply these two changes together. So 10 times 10 is 100. And so we could say that a pH of 5 is 100 times more acidic than a pH of 7. And we can do that in the other direction as 7 to, let's say we go 8, 9, 10. Let's say we change from 7 to 10. Well, that represents 1, 2, 3 units. So we have 10 times 10 times 10, which gets us 1,000. But notice that we've gone into the basic range. So we could say that a pH of 10 is 1,000 times less acidic. You're paying close attention to whether you're becoming more or less acidic. So we could say a pH of 10 is 1,000 times less acidic than 7. So we have, if we go to the right, if we go to numbers that are less than 7, we're going to become more acidic. If we go to the left, or if we go to numbers that are greater than 7, we'll become less acidic. But every time we change a unit, we're changing by 10 times. So we have a pretty quick flux in how acidic or how basic a solution is. And again, that can be potentially very dangerous. So for instance, our blood pH should be at a pH of 7.4. That should be our normal blood. If we change that to a pH of 7.8, we've only changed 0.4 units, but if we stay at a pH of 7.8 for too long, that can actually lead to seizures or even death. So our body really does not want to see these changes in pH, especially because each unit represents a 10 times difference or a 10 times change. So it's really, really important that our body has a way of making sure that our pH stays where it's supposed to stay. And that's where solutions like buffer can come into play. So if you've ever heard about buffers, you can really think of these things as pH sponges, where these buffers exist in order to resist changes in pH. Your book goes over this example here. So we have a buffer that's in our system, which helps to regulate blood pH here. So essentially what happens is when our cells are working very hard, they create carbon dioxide as a waste product. Carbon to just be floating around in our bloodstream because it can change the pH of our blood. So what happens is that when carbon dioxide enters our bloodstream, it's going to combine with water. And that's being shown kind of on the farthest left-hand side of this chemical reaction. When carbon dioxide combines with water, it creates a very weak acid called carbonic acid, which is H2CO3. So we can see that here happening in our red blood cell. Carbonic acid, when it is created, it's an acid, so because our blood is mostly water, it's going to dissociate. And it dissociates into a hydrogen ion and what is called a bicarbonate ion, HCO3-. So we see this dissociation happening here. What's important is that this is actually going to help our body regulate pH. If our blood pH becomes too basic, carbonic acid will separate into these two ions, but the reverse can also happen, that if we become too acidic, it can reform into carbonic acid. So we have this really important back and forth that's constantly happening in our bloodstream to help regulate how acidic or how basic our blood is. So really, without having to memorize this chemical formula or chemical reaction in detail, just know that buffers really work when things become too basic, they can release hydrogen ions to bring our pH back closer to a more acidic level. So they just help resist changes in pH. So that brings us really to the end of the basics of chemistry. With kind of the collection of videos we've talked about so far, we've talked about now atoms and kind of what they are. We've talked about how they make bonds. Remember, we have both covalent and ionic bonds. We then talked about water and why it is so crucial for life in that Amoeba Sisters video. And we've now talked about acids and bases and what they are and what they do, how we measure how acidic or how basic something is. What we're going to do now is move into really the last bit of chapter 4 that I'd like to discuss with, chapter 2 that I'd like to discuss with you all, which are the 4 molecules that are required for life. So again, this is a great spot to pause if you are short on time. But if you're not, let's go ahead and discuss the 4 major molecules that are required for life. So if you remember all the way back to our very first lecture, we talked about how life is composed of 4 key molecules. And we're going to talk now about what those molecules are and how they are formed. So a molecule, by definition, it's also a type of compound. Remember, compounds are created when atoms come together and create bonds. Molecules specifically refer to compounds that are made when atoms form covalent bonds. So in all molecules, the atoms that make up these compounds, they are sharing their electrons. That sharing can be unequal or equal, so we can have polar or nonpolar bonds, but either way, we are sharing electrons in this case. When you hear the term macromolecule, macro just means large. So when we refer to macromolecules, we're talking about really large molecules. They're still small in terms of like we can't really see them, but they're large in kind of the cell sense. Another term that you will hear being discussed with macromolecule is this term here, polymer. Polymer, poly means many, and mer means things. And so polymer is just a large kind of molecule that's made from many smaller subunits. And we call those subunits monomers, mono meaning one. So something to keep in mind is that for all of the macromolecules that we talk about, we're actually talking about the fact that there are these monomers, which are shown kind of in gray in this picture here. And those monomers need to be linked together in order to create a polymer that we see here at the bottom. So notice there are 3 monomers that are linked together to create this larger structure. Anytime we need to link monomers together, we perform a special reaction where basically we remove water or we dehydrate our molecule. And by dehydrating the molecule, that allows these smaller subunits to share electrons and create a larger molecule. So we're going to see that for all of the macromolecules that we talk about, each class has specific monomers or building blocks that make up the larger polymer. And the 4 macromolecules that we're going to be talking about that are absolutely required for life are the carbohydrates, which we kind of commonly refer to as sugars, lipids, which we know commonly as fats, proteins, and nucleic acids. So really the rest of the lecture is going to be looking at all these things individually. We're going to be looking at the monomers that make up these polymers, and we'll also be talking about, in general, some of kind of their main functions in the cell. So we're going to go in the order that these are listed. So let's go ahead and start with carbohydrates. So carbohydrates in general are compounds that consist of the elements carbon, hydrogen, and oxygen. And remember that is C, H, and O. And so if you look, for instance, at the chemical formula of a carbohydrate here, excuse me, we can see that really there's only carbon, hydrogen, and oxygen that makes up this molecule. Carbohydrates in general can be thought of as energy storage molecules. So their whole job is to store energy that can later be released and used by our cell to perform work. And that work can be anything. It can be anything from needing our muscles to contract or needing to transport something across a cell or out of a cell. Really anything you could think of, any job that you could think of that requires energy, carbohydrates or sugars are probably going to be used or are storing the energy for that work. Typically, carbohydrates are classified by their size. And there's 3 kind of classifications we're going to talk about, which are the monosaccharides, oligosaccharides, and polysaccharides. Saccharide means sugar. So when you see that word, saccharide, think sugar. And then the prefix in front is basically telling you how big that sugar is. Mono means 1. So in this case, in the picture that you're showing here, we are looking at the monosaccharide called glucose. And so we can really think of monosaccharides as the monomers. We're going to see that when we make these other carbohydrates, we're going to link monosaccharides together. There are lots of different types of monosaccharides, but glucose is one of the more common monosaccharides that we will talk about in this class. And it can often be drawn in different ways, where we can draw this as a straight chain, like we see here on the left that I've just starred. But commonly we draw glucose as these ringed structures here in the middle and the far right of this figure. The only difference that I want to point out between the 2 figures in the middle and the right are, notice that, so when we create these ring structures, our picture starts to get kind of messy, right? Like there's lots of C's and O's and H's and they're kind of all overlapping and it's harder to kind of see what's going on here. So oftentimes rather than draw out all of the carbons, instead those carbons are drawn as just peaks. So notice in the figure on the right, we've just removed the carbons, and instead we just see kind of like a point where where a bond is forming between that carbon. So we still have those carbons here, it's just kind of a shorthand way of making this figure look a little bit nicer in a sense. So this figure is showing the exact same thing, that glucose can form a ring, but we're just simplifying that figure a bit. So monosaccharides are very simple. Again, there are lots of different types of monosaccharides. We'll see another example of a monosaccharide on the next slide, but these are the smallest types of sugars that we'll see. And it's the monosaccharides that are truly used to retrieve energy from the cell. The other two classifications, oligosaccharides and polysaccharides, are really used to store energy. So the oligosaccharides are kind of our medium-sized carbohydrate. They also work to store energy. And oligosaccharides are made when we link monosaccharides together. So in this figure, notice we have a monosaccharide, glucose, and a different type of monosaccharide, fructose. Both of these are simple sugars, but when we link these together, we can create an oligosaccharide called sucrose. Another way that we can refer to the sucrose is as a disaccharide, di meaning two, to show that we've linked two monosaccharides together. Sucrose, this is table sugar. So this is the sugar that you use to sweeten your coffee or when you bake. So sucrose is actually or table sugar is a oligosaccharide, or kind of like medium-sized sugar. And again, it's made by linking two monosaccharides together. But we can get even bigger. We can link even more than just two monosaccharides to create our last classification, which are the polysaccharides. So the polysaccharides are huge. The two common forms, so in humans we have the form glycogen, and glycogen is really made by linking like thousands of monosaccharides together. So this picture that you're seeing here on the left-hand side, it's really just showing you kind of just a simplification of that. In this, we've only linked, like 7 monosaccharides together, but in reality, there's thousands linked together. And this is really how our body stores energy. Glycogen specifically is made and stored in the liver. And so when your blood sugar drops, for example, you have signals, hormone signals that work in your body to talk to the liver that says, hey, can you break down some glycogen and release glucose so that my blood sugar can go back up? Conversely, when our blood sugar is too high, our liver makes more glycogen. So glycogen is how we store energy. And again, we're creating a polysaccharide here because poly means many. So we have many monosaccharides linked together to create this molecule. So glycogen is always happening in animals like us, like humans. Plants, they have a similar storage molecule, but plants create what is called cellulose. It's basically almost the exact same thing as glycogen, just instead of being kind of branched like we see in this diagram here on the left, cellulose tends to be it creates these nice straight chains that we see in this figure here. Cellulose is typically stored in the cell walls of plants. We're going to talk more about what the cell wall is, but basically it's this boundary that creates this hard kind of layer outside on the surface of the plant cell and allows the cell to stay kind of stiff, and it acts almost as like the skeleton of the cell. But cellulose is the exact same as glycogen in that thousands of monosaccharides are linked together to create this storage molecule. So polysaccharides, we have many sugars linked together. In animals, the polysaccharide that we create is glycogen. In plants, the polysaccharide we create is cellulose. But in all of these cases, the mono, the oligo, and the polysaccharides, really the whole goal here is that we are storing energy. Those covalent bonds between the carbons, the hydrogen, and the oxygen or atoms within that compound, they have a lot of potential energy that could be used to do work in the cell. So our next type, so that's the carbohydrates. So let's look at the next class of macromolecules, which are the lipids, which the lipids, again, we can think of as the fats. And they have kind of a multitude of functions. They also function in energy storage, but they can also help in insulation. So this can also help regulate our body temperature. And also we're going to see makes a main component of the cell. So it helps basically create cellular structures that are very important. This class of macromolecules is very diverse. So there's lots of different types of lipids. They all look very differently, and they can each perform different jobs. But one thing that makes them kind of fall into this category is the fact that this class of macromolecule is very non-polar. What that means, so we're going to see if you look at the molecules here on the right that I've starred, notice that we still have carbon, hydrogen, and oxygen, but we really have a greater amount of carbon and hydrogen in these molecules. Carbon and hydrogen, when they bond, they create non-polar covalent bonds. So they're going to share their electrons equally. That's important because that actually makes these molecules insoluble in water. That means they can't dissolve in water. And we've seen this all. If you've ever added, like if you have a glass of water and you've poured like cooking oil on the top, you've seen that those two things stay separate, right? That's showing you that that oil cannot dissolve in the water that you've tried to add it to. But again, this is a very diverse class, and so we're going to see that these come in kind of many different types and sizes. But there's 3 lipids that I want to talk with you guys about that are very much required for life. And those are the triglycerides, the phospholipids, and the steroids. So the triglycerides, which are being shown here on the figure to the right, they get their name because they have a molecule called glycerol. And attached to that glycerol are what are called fatty acids. And there are 3 of those fatty acids. So tri for 3. So the tri is telling you that there are 3 fatty acids, 1, 2, and 3, attached to a glycerol molecule. And triglycerides really make up kind of the common fats that we use in our everyday cooking. They can make our cooking oils, so we have, like vegetable oil and olive oil. They can also make butter. So these are really important. These types of fats are going to be really important for our energy usage. We're going to use, and our body can kind of store energy in the bonds that make up these macromolecules. Notice that, so we've all experienced that some triglycerides are liquid at room temperature versus solid at room temperature. And that actually has to do with the fatty acids themselves. Notice in this picture that we have these 2 fatty acids on the, notice that these fatty acids create very straight chains. We call those saturated fatty acids. What that means is, if you notice, every single carbon in that chain is completely saturated with hydrogen. So it's bonded to either 2 hydrogens or 3 hydrogens, depending on if we're kind of in the middle of the chain or the end of the chain. When that happens, we get this really nice kind of straight chained situation that we see here. But notice in number 1, in fatty acid number 1 here, we have a situation where instead of being bonded to 2 hydrogens, carbons have made a double bond between themselves. And because of that, it kind of creates that chain to be kinked. It kind of causes it to shoot out here to the left. This type of fatty acid is called an unsaturated fatty acid. And unsaturated fatty acids, because of that kink, whether it being kinked or being straight chained, this is going to control how close together these molecules can get. So for example, think of saturated fatty acids. Imagine that we were all in a class together, and I asked you all to stand up with your arms right next to yourself, standing up very straight. And then I asked you to get as close as you could possibly get together. You'd be able to get really, really close. You'd be able to pack very tightly. When that happens in triglycerides, that causes those things to be solid at room temperature. If you can pack really close together, your molecules can get close together and you can create something like butter. So butter represents triglycerides that have those saturated fatty acids. But now imagine that I asked you to stand together but to shoot your arm out. So you have to hold your arm out at a 90 degree angle. And then I asked you to pack together. Now, if you're acting like an unsaturated fatty acid with that kink, it's going to be harder for you to pack. And so because of that, triglycerides that are mostly unsaturated tend to be liquid at room temperature. Those molecules can't get so close together, so they remain more fluid. And that's what we see with like olive oil and cooking oil, like canola oil. So just kind of a fun thing to know. Now you know why some fats are solid at room temp and why some are liquid at room temp. But again, triglycerides are really going to be your energy storage molecules. The other class of lipids that are essential for life are the phospholipids. And we're going to talk a lot about these when we get into the cell lectures next week. But really what I want to point out is that there's kind of a similar structure in that we have a glycerol molecule and we have two fatty acids attached to that glycerol molecule. But instead of a third third fatty acid, we have what I'm just going to collectively call a phosphate group attached to that third position. And this makes this molecule very unique. What actually happens is that kind of this yellow head, so phospholipids kind of look like a balloon with two strings. The balloon portion of this phospholipid is very polar. What that means is that it loves water. Another term that we can use for water loving is hydrophilic. Hydro meaning water, philic meaning lover. So this portion of our lipid actually wants to be around water. But the two tails, those fatty acids that come off of the other side, those are very non-polar. They are very hydrophobic, so they hate water. So this portion of the molecule hates water. And we're going to see that this creates a really unique situation which allows us to create what is called the plasma membrane in our cells. But we're going to talk more about that when we get into the cell itself. But essentially what's going to happen is that this molecule is going to arrange itself so that the phosphate group that loves water is going to face water, and the hydrophobic tails that hate water, they're going to hide and not be anywhere around the water environment of the body. All right, and our last category of lipids that I'd like to talk about are the steroids. And we often kind of in our everyday life hear of steroids as being bad. A lot of us talk about how bodybuilders like Arnold Schwarzenegger, your muscles big and allows you to win bodybuilding competitions. It can have some really deleterious effects on you later in life. But actually, steroids as a class of lipids are very crucial and important for human health. Many of the hormones that create our sexual differences between males and females are actually caused by steroids. For instance, estrogen and testosterone are two steroids. Both males and females express or have both of these steroids in their bodies. Males typically have a greater amount of testosterone than estrogen, which gives them kind of their male physical features. Women, on the other hand, produce more estrogen than testosterone, which causes more female sexual characteristics. So these are steroids that are very important to basically creating kind of our physical features and allowing our reproductive organs to work in the way that they're supposed to. Another steroid that sometimes gets a bad rap is cholesterol. So we talk about cholesterol being present in like kind of high fat or fried foods like onion rings, burgers, and French fries. And yes, that is true, but cholesterol is also produced naturally by our body, and it can be a really important signaling molecule, and it's also found in our cells, specifically in the membrane or the barrier of our cells, and helps kind of give stiffness to those cells. Making sure that you don't eat too much cholesterol in your diet is really important because of the fact that our body can make that on its own. So it is important to kind of make sure that you're eating healthy and not overwhelming your system. When you have too much cholesterol, that can cause like clotting in your blood, or like the hardening of your arteries and veins, which can put you more at risk for heart attacks and things like that. But these are the three types of lipids that are required for life. Triglycerides really work as energy storage molecules. The phospholipids are going to basically make a main component of our cells, and the steroids are really important for signaling and regulating like body functions in our body. Okay, so we've talked about carbohydrates, we've talked about the lipids. We have two more classes of proteins, of macromolecules to discuss. And next up are our proteins. So the proteins, again, these are large macromolecules, and their building blocks are called the amino acids. And the amino acids in general, there are 20 amino acids that are used and linked together to make a variety of proteins in our body, but they all share kind of this general structure that's shown here on the top of this figure, where all amino acids have what is called an amino group, which is a nitrogen and two hydrogens. That amino group is attached to a central carbon here. That carbon always has a hydrogen atom attached to it, and it has what's called a variable group, or R group, or side chain here. You also might sometimes see R group here. We're going to come back to that R group here in a second. On the other side of that central carbon is what is called a carboxyl group, and it's highlighted in yellow here. Carboxyl is always a carbon that is double bonded to an oxygen and then single bonded to a hydroxide or OH group. So all amino acids, everything that I'm kind of starring in blue, that's the thing that every single amino acid is identical about. But it's really this R group, or this side chain, that can vary. So this varies between each of the 20 amino acids, and that's what makes each amino acid unique. Essentially, what's going to happen is, in order to make a protein, we need to link amino acids together. So for instance, we have our first amino acid here on the bottom called glycine. You can see its central carbon, its R group is just a hydrogen. And we have our second amino acid here called alanine, and its R group is the CH3 group here. So notice they're identical except for those two R groups that I've circled in blue. But when we link these together, when we remove water, we can then create a peptide or protein. So peptide is another name for protein. But when we link amino acids together, that's what's going to create the protein. And we can link like a few amino acids or hundreds of amino acids together to make a small or large protein. But the order of amino acids matters. And so what I want to kind of focus on is actually what is the final structure that proteins make. So proteins are probably the most varied group of macromolecule that we will find in the cell. And every single protein folds into a very different 3D structure. And it's that final 3D structure which gives each protein a very specific function in the cell body. So no two proteins are the same. Each different type of protein has a very different 3D shape, which allows it to do a very different job in the cell or the body. But that 3D shape that we're going to achieve starts with the amino acids. So there are actually 4 levels of protein structure that end up leading to the final 3D shape that we see proteins achieve. And those 4 levels are called the primary, the secondary, the tertiary, and the quaternary structures. Not all proteins have quaternary structure, but they sometimes do. But all proteins have primary, secondary, and tertiary structure. So we're going to talk about what these 4 structures are referring to. So primary structure, that is the actual sequence of amino acids that is linked together. So remember, we have 20 amino acids. I'm going to shorten amino acid to AA. So we can have 20 amino acids, and those can be linked in a variety of orders. The order, or which amino acids are actually linked together, is determined by our DNA, by our genes that live in our cells. But the primary structure, the order of amino acids and how they are linked together, that is the very first level of protein structure. So in this example here, we have 8 amino acids linked together, and that makes up the primary structure of this protein. Once these amino acids are linked together, they're going to start interacting with one another, and that's what's going to create our secondary and tertiary, our second and our third levels. You might also see another way of saying primary is 1 with like a degree symbol. So primary can also be shown as 1 degree. Secondary is 2 degree, and tertiary can be 3 degree. So let's look at what the secondary and the tertiary structures are. So secondary structure, when you watched the water video from the Amoeba Sisters, they talked about hydrogen bonds. And remember that water, hydrogen bonds occur, for example, in water, because we have a slightly negative oxygen and slightly positive hydrogens because of the fact that we have polar covalent bonds and our electrons are not being shared equally. This situation can happen where we have slightly positive hydrogens. That can happen between a lot of different molecules. In secondary structure, what's going to happen is certain parts of the amino acids will create hydrogen bonds with one another, and that's shown by these red dotted lines here in these figures. So those red dots represent hydrogen bonds, where basically these partially positive hydrogens are going to be attracted to a partially negative atom in a different part of our amino acid chain. When those hydrogen bonds form, we can create 2 different shapes. And those shapes are the alpha helices, or 1 alpha helix, and beta pleated sheets. An alpha helix, so this is our alpha helix here on kind of the left-hand side of our picture. And notice that an alpha helix is really just a twisted shape. So we create kind of this twist that we see here, where a beta pleated sheet looks more like kind of these kinks. So we have kind of a zigzag shape here instead. But both of these are formed because of hydrogen bonds that are occurring between amino acids. So primary structure, we have amino acids linked together in a very specific way. Secondary structure, we have hydrogen bonds that are creating either this twisted alpha helix or this zigzagged beta pleated sheet. From here then, we can enter tertiary structure. And the tertiary structure is shown kind of in the third level here in this figure. And this is when the protein gains its final 3D shape. So again, all proteins have at least the tertiary structure. And this is determined, again, the amino acids are still interacting to create this, but now we can have lots of different interactions. Sometimes those amino acids can make covalent bonds. They can make hydrogen bonds. They can also make ionic bonds. So really all kind of 3 types of bonds we've talked about work together between those amino acids to make that final 3D shape. And now at this point, once this protein has folded into that 3D shape, we can think of that protein as functional. It can go on and kind of do whatever job it's supposed to do. However, some proteins do have this 4th level of structure that I do want to talk about. So not every single protein has this, but when it does, that 4th level is called the quaternary structure. So think quarter for 4 quarters in a game, 4 quarters make $1. So the quaternary structure represents the 4th level of protein interactions. Again, not all proteins have this level, but when proteins do have this level, what's happening is that there's actually multiple subunits or smaller proteins that are going to come together to make a larger, more complex structure. So for example, we have kind of 2 purple proteins. Those proteins have completed their tertiary shape, and we have 2 turquoise proteins. So we have 4 subunits in this example coming together to make an overall much larger structure. A common example of a protein that has quaternary structure is hemoglobin. This is a molecule that's responsible for carrying oxygen in our blood. But this is a protein that requires 4 of these subunits to come together in order for it to carry oxygen properly in our red blood cells. So all proteins, we're going to link amino acids together in a very specific way. That's determined by our genes. That's our primary structure. Those amino acids can create hydrogen bonds between each other to make secondary structure, either twisted alpha helices or zigzagged beta-pleated sheets. Amino acids can continue to interact via covalent bonds, other hydrogen bonds, ionic bonds to create that final 3D shape or tertiary structure. And then sometimes we have multiple smaller proteins coming together to make a larger protein complex, and that's our quaternary structure. Before we move into the nucleic acids, there is a specialized class of proteins that I really want to discuss because these are going to come up a lot as we go through kind of our future lectures. And that class of proteins are called the enzymes. So these enzymes what they do is they catalyze chemical reactions. What that basically means is that they speed up chemical reactions. So all of the chemical reactions that occur in our body, they would actually occur too slowly for us to survive. So we actually need enzymes. We need chemical reactions to go faster so that basically those reactions make sense for our lifespan and our daily activities. So in order to speed up reactions, we have to use enzymes. And so enzymes in this picture are kind of shown here in this like kind of yellow 3D shape that looks like it's kind of forming a little pocket or bowl. So every single chemical reaction in our body has a different enzyme that functions there. But those enzymes, because of their tertiary structure, because these are proteins, they have a very specific shape. And they basically match whatever substrate or compound they are supposed to act on. So notice in this example of a chemical reaction, we have our enzyme and we have this blue kind of compound called a substrate. And notice that those 2 things together, they fit. So notice that the substrate kind of fits into that pocket. When that happens, this enzyme substrate complex, those 2 things together, this is when the enzyme actually speeds up the reaction. So it's going to allow this reaction to happen quickly, And when that reaction happens, we're going to release what are called products. So we put substrates in, we get out products. Those products don't fit the enzyme any longer, so they're actually released from the enzyme. Notice that the enzyme goes back to kind of how it looked at the beginning. So now it can pick up more substrates and complete that chemical reaction again, over and over again. And so when we do that, we can actually build, so in this case, we're taking the substrate and kind of breaking it apart into red and blue parts. We can also use enzymes to build things. So notice in the example here on the right-hand side, we have separate substrates, red and blue. Those come together to create a complex where both of those things are now together. And notice that the enzyme looks slightly different. So note on the right-hand side, the shape here is different because each chemical reaction has a very different enzyme. But again, enzymes are really important because they speed up chemical reactions in our body. All right, so we have one more class of protein, of macromolecule that we need to discuss, and those are the nucleic acids. There are three types of nucleic acids: DNA, RNA, and ATP that we are going to talk about. In both situations, these are polymers, large molecules that are built from nucleotides. So the building blocks of these molecules on the right. We're going to talk about these here in a second, but there's two main categories of nucleic acids that you need to know about, and that's DNA and RNA. And these are probably things you've heard about a lot. We talk about DNA, that contains our genes, it contains all of the genetic information that's necessary for life. And you've probably heard of RNA a lot, specifically mRNA being discussed, for example, with the new COVID-19 vaccine. But both of these nucleic acids are made from nucleotides. And nucleotides always have a sugar, so it makes kind of this house or five-pointed kind of ring structure here. That sugar is always going to be connected to what is called a nitrogen-containing base, which is in blue here. And then always also on the other side of our sugar is a phosphate. So we're going to link nucleotides together in order to create a nucleic acid. So let's look first at RNA. RNA stands for ribonucleic acid, and notice that we have, in this case, our nucleotides. We have 4 nucleotides here that are linked together to create this simple ribonucleic acid. Notice that between these 4 nucleotides, the phosphate and the ribose sugar are always the same. So the name of the sugar here is called ribose. That's where the ribo part comes from in the name ribonucleic acid. But notice that what is different is to the right-hand side. So there are 4 different nitrogen-containing bases, adenine or A, cytosine or C, guanine, G, and uracil, U. So it's these 4 bases that make the unique nucleotides. And so RNA will always contain these 4 nucleotides, C, A, G, and U. In terms of its overall structure, RNA is always single-stranded. So we're just going to link a bunch of nucleotides together to create one long strand of RNA. DNA is a little bit different. DNA is also made from nucleotides, but those 4 nucleotides are slightly different. We still have adenine or A, cytosine C, and guanine G, but notice now thymine or T. So we have T's instead of A's. So that's really important. That's one of the big first differences between DNA and RNA. The other big difference is the sugar. So the sugar, instead of being called ribose, is called deoxyribose. DNA stands for deoxyribonucleic acid, so the deoxyribo part refers to the fact that the sugar is deoxyribose. DNA also always creates a double strand, and that double strand always looks like a twisted kind of ladder structure here, which we refer to as the double helix. So this structure here is the double helix. So DNA is always double-stranded, RNA is always single-stranded. We have T instead of U in DNA, and our sugar is deoxyribose instead of ribose sugar. DNA is really important because it carries our blueprint for life. So it carries our genes, and it basically tells everything about our body how to behave. So we have to have DNA. RNA typically works, it can do a lot of different functions, but the RNA that we'll most commonly talk about has to do with, it kind of acts like a messenger, and it basically carries smaller bits of information that allows kind of our cells to work with it rather than having this huge library that's stored in our DNA. It kind of takes out books. It acts like the books that you can then read. But we're going to talk all about that when we get into genetics at the end of the semester. All right, and finally, the last molecule that I want to talk about that kind of falls under our nucleic acids because it's made from a nucleotide is ATP, or adenosine triphosphate. Adenosine triphosphate is a adenine nucleotide. So notice kind of this portion is the same as the adenine we've seen on our previous slides. And instead of being attached, this nucleotide, instead of being attached to one phosphate, is attached to 3 phosphates. And actually, I'm going to number those in. So 1, 2, and 3 here. What's really important is that the very last bond, the bond that is between the second and the third phosphate, that bond has a ton of stored energy. And so adenosine triphosphate is really considered our energy molecule of the cell. Sugars store energy. ATP is really our usable energy. So we're going to see that anytime energy is required in a chemical reaction, ATP is going to be here. It's going to be used throughout the body in order to provide energy for reactions. Basically what happens is during a chemical reaction, this bond is going to be broken. So that high energy bond breaks. That actually causes that phosphate here. We have phosphates 1 and 2 still attached to our adenine, but phosphate 3 has fallen off. Because of that, this molecule is no longer called adenosine triphosphate, tri for 3 phosphates. It's now called adenosine diphosphate for the fact that there's only 2 phosphates attached to this molecule. ADP can be recycled back into ATP, so we can perform a chemical reaction that reattaches that third phosphate. And so really we can see in the cell that we have this cycling between ATP and ADP whenever we are using energy. All right, so that was a longer lecture. That's I think this week and maybe next are really gonna be our longer lectures of the semester. But that is what you need to know for chapters 1 and 2. And so again, you can use the practice worksheet that I provided you guys to help you kind of review this material. You can watch, there's videos on YouTube, you can reach out and schedule office hours with me. But that is the end of our chapter 2 lectures.